Alright, this topic in chapter 18 is eukaryotic gene regulation. In the last two videos, I went over operons, the two types of operons, which were a form of prokaryotic gene regulation, but those are not in eukaryotes, so this is how eukaryotes regulate their genes. So the main one that you really need to know for the test tomorrow, if you want to know anything, is DNA packaging. It's a unique process that is only used by eukaryotes for gene regulation. Um, so this is a photo of just like linear DNA. This is like the baseline. This is like not folded at all. It's completely relaxed. It's what we think of when we're looking at DNA. And the number one rule or phrase for DNA packaging is the more folding there is, the less transcription is going to occur. So you'll see what I mean here on that. So there are four levels of DNA packaging. The first level is called a nucleosome or a beads on a string model. I prefer beads on a string, it's very logical. You'll see why in a second. So basically, this gray part here, this is our DNA, and this green thing here is called a histone core or the bead. It is made up of 8 histone proteins, 2 histone 2As, 2 histone 2Bs, 2 histone 3s, and 2 histone 4s. Yeah, I don't think we really need to know this, but she mentioned it in class, thought I'd mention it here. But that is this histone core. So what happens is our DNA, I'm sorry, the DNA is not the gray thing. The DNA is this red. So our DNA is this red string here. So what happens is the DNA loops around the bead twice, and each bead is 10 nanometers in diameter. So it makes it much, much thicker, and the strand itself much shorter because think about it like physically, you're physically wrapping it around this bead. And you do that with lots and lots of histones. And what the H1 histone protein does is it holds the DNA onto this bead and keeps it in place. And it stays in place with the H bonds as well as the H1 histone. And the reason that this controls gene regulation or regulates genes is because the promoters on this DNA here, they're so tightly bound up that these genes that they code for physically cannot be coded for because the RNA polymerase physically cannot bind to that DNA. And then down here we have a bigger zoomed out picture on an SEM micrograph where we can see all of these beads and on the DNA and how thick it is. As you can see, 100 nanometers, but each bead is 10. So it's skinny, but it's much thicker than just a string of DNA. Now, level 2 is pretty simple, I would say. What happens here we have our beads on a string model right here. This is our beads on a string. And you can't see me, but imagine in this image, you're physically wrapping it up even tighter, 3 times thicker. You're stacking 3 of these beads on top of each other to form kind of like a rope of DNA. And it's 30 nanometers. The last one was 10 nanometers thick. It says 11 in this picture, but it's 10. Um, and now we have our 30 nanometers thick. So you're tripling the thickness and the folding of this DNA in one level. And because that even more H bonds form, it gets even thicker, and even less transcription is occurring for the same reason. The RNA polymerases are getting more and more hidden away, condensed. The RNA polymerase can't get in there to the promoters, so it's just even more difficult for those genes to be turned on. Level 3, now we're in like the very, very condensed part. So this blue here, that is our 30 nanometer chromatin fiber. This here is that blue. And it is called a loop domain. And it's going up and up and up, and it's getting all looped up even more. And how we keep it attached is we have this protein scaffold on the bottom. So we have all of our 30 nanometer chromatin fiber getting looped up and looped up and looped up and looped up. And then we attach it here to the scaffold. And that's just to keep it in this shape, keep it all folded up. And like I said, this is like one of the higher levels. We're almost at level 4. and we're at level 4. Level 4 is called a metaphase chromosome. So what happens here is here's our um oh my gosh, what is it called? Scaffold. Here's our scaffold, our loop domain here, and we have it. Instead of bending the DNA, we're going to bend the physical scaffold into this V shape up and down, up and down, up and down. And this is like the highest level, and it's so folded that absolutely no transcription is possible. All of the promoters are so coiled up and hidden that no RNA polymerase can get in there at all. None of these genes can be transcribed. And some terms and things to know about DNA packaging. Euchromatin is what we call open and relaxed chromatin, and so there's high levels of transcription. So if we go back to our original photo, this would be a good example of euchromatin, but even still, beads on a string would still be a good example of it. While there still is some packaging going on, it's still pretty available to be these genes here. There's still lots of genes available that can be transcribed and still high levels occurring. But then the opposite of that is heterochromatin, and that is tightly bound chromatin, level 3 and 4, like I mentioned, and there's very low transcription, and in the case of level 4, absolutely none occurring at all. And then some more things to know. Within one single chromosome, the packaging levels can change. So, for example, if I have a muscle cell, the gene that codes for hair, obviously we don't need that. So that might be a gene that we have folded at level 4 because we don't need it in that cell in the muscle cell. But then for the same thing, they can be loose, all sorts of levels within one chromosome. But the centromere, which is the center of the chromosome, I didn't put a picture because I hope we can all envision that. It's the center of the chromosome and it's always highly coiled. So any genes close to the centromere are never used within that cell because of how close they are. It's so highly coiled that those can't be used at all. So those genes are just always inactive in those cells. That was kind of what we saw on our problem set, one of the thinking critically problems. This is why those genes weren't being activated. And then we have two more um, additional ways that eukaryote cells regulate their gene expression. These two are important to know, but the packaging is just such a bigger concept, and like it's something you need to know more than these ones, but still understand these as well. So DNA methylation, DNA is a polar molecule, as we know because it is polar, and a methyl group here I have up on the screen. This is a nonpolar I think amino acid group. I forget what they're called exactly, but a nonpolar group. And what we do in DNA methylation is we attach one of these methyl groups to the DNA. And because it's nonpolar and DNA is polar, that's a big difference. So RNA polymerase can't bind to that nonpolar DNA, that promoter, so transcription greatly lowers. So if we want to turn off a gene without actually folding it, this is a way to like chemically stop a gene from being transcribed. And then basically the opposite of that is histone acetylation. Um, an acetyl group on the screen here, this is a polar group. So what we do in histone acetylation is we attach this acetyl group onto the DNA, and that keeps the DNA open because it repels the histone proteins from packaging the DNA, as we saw back all the way back here. It basically, adding the acetyl groups repels this histone proteins from even coming on to the DNA in the first place, and this causes the transcription rates to increase. So overall, the biggest thing to know from this eukaryote gene regulation, in my opinion, is the DNA packaging, just because it's it's something you it's something you need to know. It's within chromosomes and with all eukaryote cells. The other two are important. Like I've already said this before within this video, but yeah, I would focus on this, reviewing this, and looking up pictures, looking in the textbook just to see the pictures and understand it. And also just to know that operons are not eukaryote. gene regulation. Whatever you do, know that operons are not eukaryote gene regulations. That's something you do not want to mix up. So if an FRQ is talking about describe or draw or something about eukaryote gene regulation, do not draw an operon. That is in prokaryotes, bacteria only, and you need to separate those two things in your mind and understand that they are not the same. That's, I think, the biggest thing that you need to know, the difference between those. And yeah, hopefully this was helpful. I try to get some good pictures in here. Yeah, let me know if you have any questions in the comments, and good luck on the test tomorrow and on Friday.